Thanks very much, Ashley, for the introduction, and I'm glad to be able to present a little bit. I'd like to set this more in an aging context, given the conference. So first, I'd just like to show some analyses that we did of prevalence of cognitive decline. So there's an old study done in the UK, an MRC CFAS study in 13,000 subjects in primary care. And you can calculate from the data the by age proportion of people who are below any given mini-mental cut. Mini-mental is simply a 30-point scale that measures cognitive decline. So you can see at mini-mental cut 26, mini-mental cut 20, and so on. You can figure out the prevalence. I was able to get the same thing because I had access to some Chinese data collected between 1998 and 2012 in 5 health regions of China. And from the data, you can do the same sort of calculation. The most extraordinary thing was that when you put the two together, they're pretty well identical. Different time, different culture, and yet there's something fundamental going on here, which is not particularly linked to culture. And this is just a crude instrument like mini-mental score. So what might that underlying big aging substrate be? So we've already had from Dr. Sinukson an introduction to tau protein. I don't have to repeat. So it's basically a microtubule stabilizing protein that's critical in the functioning of the corticocortical projection system mediated by pyramidal cells, and tau protein aggregates in Alzheimer's disease. The tangle is made up of paired helical filaments, and the structure is shown on your right there. It's a polymer of repeating C-shaped subunits. People often think that phosphorylation is somehow critical. When we analyze this in detail, less than 5% of the tau that is in paired helical filaments is phosphorylated. And even that, it's only on the surface or the coating and is not part of the proteolytically stable core of the filaments. When we first isolated tau from the core of the paired helical filament, you can see the repeat domain up there on the right, and the orange segments represent the 3 classes of fragments that we isolated from the core of the paired helical filament. They all come from the repeat domain, but they all map to exactly a 3-repeat segment, even though they come from either a 3-repeat isoform or a 4-repeat isoform. And that segment represents exactly 3 repeats in length, but it is, if you look down the left, bottom left, it's out of sync with the tubulin binding linker segments of the molecule. The repeat domain is constructed out of alternating binder segments, tubulin binding segments in black, and linker segments. Well, you have in the paired helical filament exactly that configuration, but out of sync by half a repeat. Now, we reproduced that precise out-of-phaseness by binding tau in vitro, and we're able to demonstrate a characteristic GLU391 truncation, which localized the C-terminus, and we knew the N-terminus from N-terminal sequence analysis. What that showed was that this fragment is essentially the propagating thing, the element that propagates tau aggregation. And it causes this conformational flip in the repeat domain that is self-catalyzing and self-propagating, so that you always end up with more aggregated tau that is bound in this altered conformation. And the word that's used for that, which I'm describing in too many words, is prion-like processing. So tau is a undergoes prototypic prion-like processing. And as we now know, that these oligomers, once they form, are infectious and spread throughout the brain. Now, that spread is very characteristically defined by Braak staging. And it's this very characteristic spread out of the medial temporal lobe structures into the temporal into isocortex according to the 6 Braak stages. Braak stage is highly correlated with cognitive decline measured in the 12 to 18 months before death, and this was just one of the studies. This one happened to be prospective and in an epidemiological context. In fact, part of the CFAS studies that I alluded to before. And you can see this very close link between Braak stage and mini-mental decline, and also on your left, the same sorts of correlations between Braak stage and decline in functional imaging, as shown in this case by SPECT, but you can see the same thing by PET. More recently, the same sort of correlations have been demonstrated during life using ligands for aggregated tau, in this case the Tohoku University compound, but there are a couple of them in development. The Lilly compound produces a similar sort of picture. Again, I'd like to sort of use this bit of tauology to go back to the aging theme of the conference and show you some calculations of the prevalence of tau pathology. So on the left there, you have the age-specific probabilities of Braak stage transitions. The yellow is from nothing to Braak 1 or beyond. Green is from 0 or 1 to 2 or beyond, or 3 or beyond, and so on. So they're just probabilities of transitioning by age. And this comes from a big case series that Braak's group published, and based on 847 cases post-mortem. You can apply those probabilities, or really prevalences, age-specific prevalences, to the US population for 2010 and calculate, okay, well, what is the age by Braak stage of the US population? Oh, sorry, yes, by Braak stage. So the yellows, there's 31 million people in the US who are at Braak stage 1, 13 million who are at Braak 2, 12 million who are at Braak 3, 7 million who are at Braak 4 or beyond. It's really only the 4 or beyond, or the 7 million, or actually watered down to 5 million, that appears as the usual Alzheimer's disease prevalent statistic for the US. But there's all this other stuff going on beforehand. The second important thing is you can be at Braak 1, but in this cross-sectional analysis at least, and not necessarily progress to Braak 2. But the number at Braak 2 ends up being the number at Braak 3. In other words, once you're at Braak 2, that's it, you're done. Now, this obviously has to be done longitudinally, and it's very hard to do, and maybe when we have the imaging ligands, we'll be able to get a better sense of this. But the bottom line is really scary, that if you consider the over 45 population in the US, 50% have some sort of tau pathology. Remember, we're talking about an endogenously generated infectious process that is slowly spreading around the brain at about 10 years per Braak stage. So it's this slow burn progression. Of that roughly 50%, 25 are at Braak 1, about 10 are at Braak 2, about 10 are at Braak 3, and 5 are at Braak 4 or beyond. So that's the picture of tau pathology in the brains of Americans. And you can figure out, if you're in a clinic and seeing Mrs. 75-year-old or 72.5-year-old, you can simply calculate what's the probability that she's got no tau pathology in the brain. And if you look at the 72.5, in other words, it's the middle of the 70 to 75 age group, you can see that her probability of being at Braak 1 is a Braak 0, in other words, having no tau pathology, is only 30%. She has 70% probability at being at one of the Braak stages. Now, you can see how this progresses over time. So you can see in 2010, there are 63 million people affected. In 2030, it's 88. And in 2050, it's 105. These are simply from WHO projection figures doing the same probability calculation. What's happening societally is this huge increase in the dark blue in the BRCA4 or beyonds. Same thing happens in Europe, essentially the same dynamic going on. In Asia, it looks more benign, so to speak, simply because generally across Asia the populations are younger, but they're aging. But on the other hand, the numbers are enormous. So today, there's 500 million people across Asia who have some degree of tau pathology in their brains, and that will go up to 1.2 billion by 2050. So the implications of this kind of BRCA-ish epidemiological analysis, I said this is a self-propagating pathology, extremely widespread, and the numbers are truly staggering. There's 335 million people in Europe, Asia, and US who are at BRCA stage 2 or beyond today, and that's going to go up to 860 million people. So whether you're a Taoist or not, it seems that it's an important problem to tackle and to target. So how does it begin? And here I'd like to really link up somewhat with the previous talk in a slightly different angle, but similar. When we got these really pure paired helical filament preparations that enabled us to isolate this short tau prion fragment, we found co-purifying 3 mitochondrial membrane proteins. And you can see the sequences that we got, and they're co-eluting, so tau with porin, tau with core protein 2 of complex 3, and tau with ATP synthase subunit 9. Now, where does all this stuff happen? Well, we heard a moment ago about these early aggregates forming in the endosomal lysosomal processing pathway. In the old pathology, this used to be called granulovacuolar degeneration, but it's really a tau storage, lysosomal storage disease. It's a stage of the pathology when it is predominantly in lysosomes. And you can see the co-localization of the tau and core 2 or tau and porin inside the lysosomes. Now, why is that important? Well, a large body of work has shown that a critical driver for initiating tau aggregation is setting a seed. And what is that seed? Well, the most likely seed comes from failed mitochondrial turnover. Mitochondria turnover at a phenomenal rate, 5 to 10% per week of your mitochondrial mass. And the endosomal lysosomal processing pathway is stressed as you age. So this, at least from my point of view, is the fundamental aging driver. What does amyloid do in this? Well, it just makes a bad situation worse because amyloid obligatorily gets processed through the endosomal lysosomal processing pathway. So most likely what having amyloid pathological mutations does is simply brings forward the fundamental age of risk, but the fundamental age thing is going on. Okay, so given that analysis, I'd just like to summarize where we are. So that's the sort of the background and origins of this, where we are with our clinical development program based on this theoretical construct. And the essence of the construct is that the tau aggregation process is driven by an age-related abnormality in endosomal lysosomal processing, that tau aggregation is driving the cognitive decline and is highly correlated with it. So the drug that we found by chance initially, methylthioninium, its target is the aggregates themselves. We identified it because it simply dissolved paired helical filaments isolated from the brain. So this shows you a series at increasing concentrations, and it turns what is this paracrystalline array into monomers which can be cleared readily by the cell, and we've shown that in cell models, which I won't go into here. How it works is probably by binding to charge-stabilized a region in the anti-parallel aggregation of tau where complementary charges stabilize the beta-pleated sheet structure. Adding methylthioninium destabilizes that cage-like structure and leads to unzipping of the beta sheet. And you can see the dotted lines are essentially the bonds that hold the beta sheet together. This is a model that comes from essentially a minimum energy model, molecular dynamic modeling. It's very hard to show this. It's very hard to show the action of a disaggregator in proper X-ray crystallography, because by definition you're destroying your target. So we did this trial some years ago now in 321 subjects, and we showed a significant effect. The primary endpoint was at 24 weeks, and we showed an effect in moderate at 24 weeks on 3 different outcome measures, on ADAS-Cog, on CIGIC, and on Mini-Mental. All of these were significant nominally, but more importantly were significant after correction for multiple comparisons, so they were quite robust. So the study succeeded in identifying 138 milligrams per day as the minimum effective dose. We had a problem with the high dose, 228 milligrams per day, and that's really due to problems with formulation and dose-dependent fluid impairment, which I'll say a few words about. There was no effect in milds because over 24 weeks there was no placebo decline in milds, so no possibility of demonstrating an effect. However, in milds we saw a very robust brain imaging effect. This was by SPECT scan, and you can see in 4 brain regions the placebo decline over 6 months, most marked in temporal lobes, and you can see that decline is stopped or even reversed at the 138 milligram per day dose. And when you do statistical parametric mapping, the regions at which the effect, that is prevention of decline, mapped to is the medial temporal lobe structures, which is where the tau aggregation pathology is. We're in the process now of reporting a large series of studies on this, and we show that the steady-state trough brain concentration of MT on a repeat dose that's giving 138 milligrams per day and 3 divided doses per day is 0.18 micromolar. The P50 for dissolution of paired helical filaments isolated from the brain is 0.16 micromolar, and in a newly developed tau aggregation assay that we're in the process of publishing, we've shown that the Ki for this is 0.12 micromolar. So it all stacks up. So it is happening in the tau regions of the brain, and the concentrations needed to do it, to block tau aggregation, are precisely the concentrations equivalent to the minimum effective dose. Most pleasingly, I showed you that there was no clinical effect in milds over 24 weeks, but there was a clinical effect in milds if you followed them longer to 50 weeks. So if you just picture this, you have the milds, there's no decline in them clinically, but their brain function goes down as measured by SPECT scan in this case. It turns out that, and the drug stopped that effect, I showed that a moment ago, the drug stopped that decline. The magnitude of that effect on the brain scan, which had no clinical correlate at 24 weeks, was highly predictive of the clinical effect that you saw 6 months later at 50 weeks. In other words, the drug is producing an effect at 6 months that you can only see clinically in moderates, but it is producing an effect in milds in the brain that you can't see clinically, most likely because of cognitive reserve, and that effect is then predicting the clinical effect down the track, which is a very pleasing result because it suggests that we could use this in the future as a surrogate for treatment response. Okay, so we, in going forward to phase 3, we've had to clean up the whole MTC problem. And we've developed a new molecular entity which we call LMTX, which is on your right, which is a stabilized, reduced form of the active MT moiety. The red things there are the dihydromesylate salts, which are linked to it. It's a distinct molecular entity from MT, but it ends up being the same thing in the body. Here you can see the data showing the dissolution defect of the capsules used in the phase 2 trial, and here you can see the food effect, that there's a dose-dependent impairment of absorption of MT when you try and take it with food. When you put these two things together, you get a very simple dose response, so that the dose available within 60 minutes predicts the full range of dose response on, in fact, all of the parameters: clinical at 24 weeks, imaging at 24 weeks, and clinical at 50 weeks. This is all down to the complex processing that the MT plus moiety has to undergo when it's absorbed in the active form. It has to be in the stomach long enough, as you can see on the right there, to be converted from the MT plus, which is charged form, to LMT, which is uncharged, reduced. And that just gets in by passive diffusion. So the absorption of the MT plus moiety is quite complicated and has taken quite a lot of work to figure out. But we've now finished that, and we're putting that out into the public domain. LMTX is much simpler. It's simply absorbed by passive diffusion. So going forward, we're now running 3 big global clinical trials. One, TauRX 015 in mild, moderate AD. We're testing 2 doses, 150 and 250 milligrams per day of MT. Another is the mild study, TauRX 005, in 700 subjects, and that's testing only the 200 milligram dose, and we're running a small study in frontotemporal dementia as an orphan indication at 200 milligrams per day. And it's the usual endpoints, which I won't go into. What's difficult in trying to design a phase 3 trial for a drug that is disease modifying, that is, that alters the rate of progression or the trajectory of the disease, is to figure out what the effect size is going to be. And it's governed by 2 random variables. One is the uncertainty associated with the effect size. And so here you can see we've used the standard FDA required analysis, a mixed effects analysis with unstructured correlation matrix, the works applied to our phase 2 data. And you can look at the effect size there and the placebo decline, but the number that really matters is the effect size as a percentage of placebo decline and its standard error. So there are 2 random variables. One is uncertainty about the effect size, and then the second uncertainty is, well, what the hell is the placebo decline going to be? And this shows you all of the studies that we could locate in the public domain, and it's a very uncertain matter what the placebo decline is going to be in the next phase 3 study that we do or anyone else does. You can see the bapineuzumab and solanezumab studies. What's frightening is the Critical Path Institute data set is even lower. What that all means is that a drug that has a percentage effect on the placebo decline has a smaller absolute effect size. We've developed a method for calculating all of this, and this shows you what we call the absolute probability, and it shows the numbers going around the curve are percentage declines with respect to placebo, so they are effectively effect sizes. On the vertical axis, you've got the probability that that effect size will actually occur, and on the bottom, you've got, for a trial size of 700, that the trial will be successful, taking account of all of that. So it ends up being that you have to get about a 25% effect for an N of 700 to have a statistically significant effect, given the uncertainty in placebo decline and given the uncertainty in your effect size. So this study is now going in all of these places, 250 centers globally in 22 countries. The current status of the total 640 subjects that it's a little bit over what we were aiming to, so it ends up being 1640. In the mild moderate study, that's completed now. We're no longer recruiting to that. The mild study, we're already over the 700 mark, and it'll stop recruiting in September, or stop screening, and then there's a 1 month before people get in. The frontotemporal dementia study, even though it's being run in 72 centers and is probably the largest frontotemporal dementia study anybody's ever done anywhere, still very hard to recruit. We've got 110 so far, and we hope to have that filled up by December. We expect the first top-line data in first quarter of 2016. Just it was planned that LMTX should have a better profile than MTC, and it certainly does, at least looking at the crude measure of withdrawal rates. The green stars are the time-specific withdrawal rates with MTC. As you can see, it only went out to 50 weeks, and you can see that that there was really quite a high withdrawal rate, and we've got a much lower withdrawal rate from LMTX. So the drug is definitely better tolerated and definitely better absorbed than MTC. And hopefully we've overcome the problems that we saw with the MTC or methylene blue that we used in phase 2. So here we are. These are the 3 ongoing studies. Probably the mild study will take a little bit longer, but the frontotemporal dementia and the mild moderate will read out more or less together in second quarter of 2016. LMTX is intended to be first-in-class tau-based treatment for mild moderate AD, and I hope I've been able to persuade you that it has a potential utility preclinically in the prevention of AD, because the roots of the tau pathology and their temporal antecedents are at least 20 years before they become evident as the clinically linked tau pathology that we see after Braak stage 4 or beyond. Thank you.